My apologies in advance. As I talk about how higher bilaterian animals should be classified, I find this topic extremely frustrating, as anyone who works with me will gladly attest, and many of my students as well. But perhaps this is also a good opportunity to demonstrate how science works. Scientists don't know how the natural world works. They simply can make hypotheses and then look at what the evidence supports. And it sometimes turns out that later hypotheses get evidence that points in a different direction. And then there seems to be a contradiction. This model is supported by this evidence. This model is supported by this evidence. That prompts more studies, more research, more data. And the idea is that gradually science gets a more and more accurate idea of how the natural world works. But scientists never at any point say, I know the answer, this is 100% true. It is a scientist's job to always doubt, to question, to look at new alternatives. Also, it should be emphasized that those who study the natural world realize that the answers aren't always clear. Outside science, I think there could be the idea, well, just do a test, get the data, and then we'll know. There are questions which have been investigated for years, for decades, and there's data pointing in lots of different directions. And one example of this is, how should we classify the higher animals, especially the invertebrates, into real, meaningful biological groups? Before I go into new material, something I stated in a previous video in this series meant for General Biology 2 students, is that if you look at segmentation, segmentation is the plan where you just kind of copy and paste body regions so that you don't have to continually reinvent things. You say, oh, now that we know how to make a rib, let's just make a series of those. Now that we know how to make blocks of connective tissue and muscle known as somites, why don't we just continue this? And so when you look at vertebrates, we're segmented, but so are chordates, and so are segmented worms, annelids, so are arthropods. And so this is important. So a question could be then asked, when in the animal family tree did segmentation arise? And it turns out that's hard to answer. And there have been different ways of trying to make a family tree of animals. In an older one which was the main one when I was in school, it was held that as animal groups slowly evolve, you had simple ones like flatworms, which aren't segmented, nematodes, which aren't segmented, etc. And then when we look here, we say, oh, the vertebrates and the chordates, they're segmented. Annelids, arthropods are segmented, and mollusks mostly aren't segmented, but the first ones were, and some still are today. So segmentation probably evolved here. Right? And it's a shared feature among these higher animals. And that makes sense because if you look at how human embryos and vertebrate embryos say, let's organize our segments, they're very often the same genes as, say, an arthropod would use to establish segments in its embryo. That's strong support for this evolving once here. These animals down here, they have no trace of segmentation. Now, as genetic studies continue, it seems that most of the genetic evidence supports a different organization of the animal groups, so that nematodes are put in the same group as arthropods. Annelids and mollusks are put in the same group as most flatworms. Not all flatworms, but most flatworms. But then let's get back to segmentation. What does that mean for segmentation? Because they're segmented, and they're segmented, and they can use the same genes. The odds of the same genetic mechanism evolving using the same genes independently in multiple lineages, that's difficult to imagine the odds. But then the only way that this would then work is then if these other organisms had segments in their ancestors, or perhaps the precursors of segments, and then lost them. So flatworms aren't segmented, but their relatives are. Nematodes aren't segmented, but their relatives are. And so The only other explanation is that this common ancestor had at least precursors of segmentation that arthropods, annelids, mollusks, and vertebrates get, but that other lineages lose, I guess. It is so much more involved than that. To start adding additional layers on here, I'd like to introduce another feature known as the coelom. The coelom is a body cavity. And the question is, some organisms are solid all throughout, all right? Other organisms are not. And it was once thought that those which have a coelom, a body cavity, they would then be related. And so that flatworms and nematodes which lack a coelom would be less closely related to the animals which possess a coelom, which would include mollusks, annelids, flies. I'm sorry that it's kind of stalling, zooming in videos sometimes don't get along well. And so here you can see in a chordate, there is a coelom. Notice there's a space. All right, the animal is not solid all throughout. And there's an advantage to this. As body parts form, organs can then expand into this space. If one were to eat a big meal, the stomach can distend without crushing nearby organs. So there's a lot of advantages to having a coelom, a space inside the body. We humans do. So the heart is not fused to the lungs. The lungs are not fused to the thoracic wall. They are in a space, an abdominopelvic body cavity. There's a pericardial cavity, pleural cavities, etc. And because there's a space now, organs can expand without crushing neighboring structures. So coelomates then have a coelom, and it was once thought that that was a trait which would then be used as classification to show, those forms which are related. The coelom does, many other things in addition to cushioning organs and allowing them to expand. If there's a fluid here, then as the body moves, like maybe a worm is causing contractions, this can actually distribute things throughout the body. White blood cells here, or the equivalent in invertebrates, can move throughout the coelomic body cavity and protect from infection. So there's a lot of value here. So notice that here, these intestines are not attached to this muscle. There is a space, so the intestines can move. They are not fused to the surrounding tissues. They are in a body cavity. Now, before we put all animals with a coelom in the same group, as I'll point out in the next couple of videos, this segmented worm has a coelom lined by mesoderm. This chordate has a coelom lined by mesoderm. That's a third embryonic tissue. Mesoderm forms most of our, say, bone and muscle. But they form in different ways. So they share a coelom. That could be a shared trait, which shows that they're related. But since the coelom forms in different ways, maybe that's not a shared trait. Maybe that's showing their independence, how they're not related. Maybe if they were related, their coelom would form in the same way. Here's a nematode, which has a pseudocoelom, which isn't lined entirely by mesoderm. And so here's an important feature. And there is the temptation to say, let's take the animals which have this shared feature and classify them together as a group. But maybe that isn't the best example of a trait which could be used to define a biological group. So just repeating what I had said, if one were to then look at how the coelom forms, it turns out that there are different ways. So if the mesoderm is given here in red, so let's say there's ectoderm on the outside, endoderm in yellow on the inside, here's mesoderm in red, that coelom developed inside the mesoderm. So notice that just within the mesoderm, cells started to move away from each other and created a space. This is known as a schizocoelom. as mesodermal cells simply spread away from each other. And so things like arthropods or mollusks, they have this type of coelom. It's a true coelom lined by mesoderm, and it forms as mesodermal cells separate from each other. But deuterostomes, such as the vertebrates, our coelom forms, it is still entirely lined by mesoderm, but it forms as an outpocket of the embryonic gut, the archenteron. And so therefore, while there are coeloms, they form in two different ways. Nematodes, they have what's called a pseudocoelom, which forms between the mesoderm and the endoderm. So thus it is not entirely lined by mesoderm. So then the question is, when did these animals evolve their coelom? Well, obviously that's not entirely clear now, because maybe this isn't one single structure. Maybe this is multiple structures which aren't truly homologous that evolved multiple times. So here's a nematode. It would have a pseudocoelom, not a true coelom. But now it becomes more complicated if one remembers, if we reorganize the way that these bilaterally symmetrical animals are classified based on genetic information. That means that this arthropod has a true coelom, a schizocoelom, as does this annelid and mollusk. So these three share that true coelom. But nematodes have a pseudocoelom, all right, and then flatworms are acoelomate. So what does that mean? Does that mean that a schizocoelom evolved different times, one for this lineage, one for that lineage there, or maybe here? Or did these groups once have a coelom but then lose it? So that makes this more difficult now that the family tree, which is created based on genetic sequences, the anatomical characteristics don't line up as neatly as they had previously. Once again, flatworms, like these planaria, they do not have a coelom. And once it was thought that that was a primitive feature, the first animals didn't have a coelom and flatworms were primitive, and the absence of a coelom was evidence of that. But now that we've reorganized this classification scheme and flatworms are the cousins of animals which have a coelom, what does that mean? All right, did their lineage branch off before a coelom evolved? Or did they have a coelom and lose it and have no genetic trace of it? This then once again is complicated. One of the things which is of great interest is the comparison of genes, and I'll be getting to this. And so, as one compares, human genes to fruit flies, especially a group called the Hox genes, etc., which I'll get to in just a minute, this could be helpful. So genetic sequences, they will be used. Anatomical sequences, they will be used. But as I'll argue, neither are 100% perfect, where one could say, here's the conclusion and now there is no doubt. One could ask, well, could we look at fossils to try to determine what the sequence of the evolution of these groups were? And the answer is, the fossils are interesting and useful. But if we're imagining that, once upon a time there was the last common ancestor of all animals, the last common ancestor of the metazoan animals, which say have a nervous system and true muscle, the last common ancestor of the entire indicating some unity similarities. What we did is we're going to be bringing down family tree that what do the fossils show? When we look at the fossils, there's often room for disagreement. Is that an arthropod? Is that any kind of anything? They have segments. One would be tempted to call them segmented worms. But others have said maybe just like a hold fast or like a jellyfish-like thing anchored to the bottom. So the point is, a lot of these fossils, we could argue a little bit. They all seem to arise in what was known as the Ediacaran period prior to the Cambrian period. So starting maybe 630 million years ago, that's when the early animals get their start. They tend to be soft-bodied and very simple, and thus interpretation is difficult and not always clear. After the Ediacaran period, animal groups, they undergo what's called the Cambrian explosion, where they become larger, more diverse. And as you ask, why did animals get such a big boost at that time period, 540 million years ago starting? Well, maybe lots of reasons. That oxygen levels were going up, that as carnivores arose, that now this encouraged new adaptations in their prey, that there were exoskeletons and shelled animals, and maybe this allowed for more types of things. Maybe new lifestyles were possible once a coelom evolved, or a heart and limbs evolved. Maybe genetic things like the amplification of Hox clusters allowed for greater complexity. And so this could have led to a couple of, had been due to a couple of reasons. Now, as I start to wrap up, I'd like to now look at the genetic evidence, how genes are compared, and then some possible problems. And then I'd like to look at the anatomical conundrum, which then results. This is a simplified version. So how could one ask genetic sequences to solve this question? Well, let's say that the last common ancestor of the metazoans had this DNA sequence that would get passed down with possible modifications to all of the descendants of this metazoan last common ancestor. Well, one lineage would lead to, say, Cnidarians, but before the branching of the bilaterians, maybe a new mutation had been developed, which then could be passed down to all of the descendants of the bilaterians. So notice that this green block exists in all of the descendants from this point, but not in this one. And then the sponge lineage could get a couple new pink mutations that only they would have. The simple flatworm lineage could get a few new mutations that only they have. That these nephrozoans would share a blue sequence, but the protostomes would then have a green sequence. The deuterostomes would have a yellow sequence. And then one could say, just count, what is the sum of the differences between these lineages, and then use the degree of similarity to calculate relationships. In general, that works well, especially if you're comparing genes which have undergone the same relative amount of natural selection, because they're performing similar functions in all of the members being compared. But there are issues to be taken into consideration. This is not necessarily as straightforward as one might think. Once again, that's one of the reasons to try to seek out scenarios where the genes you're comparing do similar functions in all members, which would minimize natural selection. So let's take the nematodes. Nematodes are now thought to be related to arthropods, but for a long time they were thought to be distantly related. Why? Well, because nematode genomes have changed more quickly than arthropods, and they've accumulated all of these red sequences that only nematodes have. And there's something known as long branch attraction. If a whole lot of new mutations are building up in this one nematode lineage, by dumb luck, you might get one that matches one of these flatworms, all right? It isn't the original mutation that they have, it just was a subsequent mutation, since mutations are occurring frequently in this. And because they are so different from arthropods, look, they have all of these red sequences that the arthropods don't have, and they have one that the flatworms have. This would then cause, whatever computer was compiling the data to push these two together, where it's not necessarily that they're the most closely related, it's just that the genetic sequences have not changed at the same rate. So if you're going to compare the rate at which new sequences are fixed in the population, if one of the branches is evolving very quickly, then that's going to change your analysis. And it turns out that there are some things that you might need to take into consideration. So size might be a factor, which could then affect number of generations per year, et cetera. Nematodes, many of them are microscopic. And so if you're comparing, these two, maybe the small size and rapid generation time of nematodes would then cause you to have erroneous conclusions. The rate of speciation, metabolic rate, which would affect, say the production of mutations and mutagen metabolism in the body. If some are parasites, all right, and so the flatworms are so different from what their ancestors might be if the new classification model is true. And maybe the early planarian ancestors were adapting to a parasitic lifestyle. Maybe that is one of the reasons. So the big point is, it is great to use genetic sequences to try to answer these questions. But there are complicating factors. It's not necessarily straightforward, and you have to take these into account. And so even if you say, oh, this study showed this, true, but once again, there could be other factors which are influencing the conclusion. Before I leave genetics, very often Hox clusters and other things are used. So I'd like to just define those. Embryos need to regulate their genes and build a body. How can you build a body? Well, one way is to say, all right, these genes will only be expressed here in the brain or here in the stomach. And so therefore, a lot of genes which are for transcription factors. These are proteins which turn on other genes. They're sometimes called the master switches that could say, all right, let's turn on the genes that, like build a brain here. One set of these master switches are called the homeodomains, and there's a very important group of these called the homeobox genes or Hox genes. They were first discovered in fruit flies. And in one of them, which we'll see in just, so there are a number which are just important periods. So there was a homeodomain gene called BAR, which then when mutated causes this slit eye on a fruit fly, which normally has a round eye. So homeodomain genes, they're important. And clearly, since flies are using homeodomain genes in their eye formation, as are humans, that some of these are inherited from early animal ancestors. But more important, somewhere along the way, a number of these homeodomain genes became located in a row. All right, and this has become very important. They were first discovered in fruit flies, these Hox cluster genes. So here's an example. If you ask, what's going on about these fly antennae? Why do they look so weird? The answer is because they think these cells don't know that they're in the head. They think they're in the thorax, and they're trying to form a leg. That's actually a leg coming out the head. So the gene is called antennapedia. There are a couple of examples. Why are the cells making a leg? Because genes in an embryo have one role to say, you are here, cell. And once you know that you're here, then you have a job to make this body part. But if you mutate that or you change that, and the cell now gets the wrong information, even though it's here, if it's told that it's here, it could make the wrong body part based on this erroneous information. And so Hox cluster genes are so very important. And in humans, there are 4 sets of them. There's the Hox A cluster, the Hox B cluster, etc., on 4 different chromosomes. One of the fascinating things about the Hox clusters is that all of these genes occur in a row, and they are expressed in that order. So when you look at the sequence of Hox genes, that some are expressed in the head, in the middle, in the abdomen, in the same order as they occur in the Hox cluster. So it's a little beyond, say general biology classes, but one of the interesting questions is, how did animals develop the ability to build complex bodies. One set of genes are not only these homeodomain and other transcription factors, which are master switches, but a series of them in a row, which then allowed for a body axis for cells to express different ones in a row. And the genes which help to program the body axis of fruit flies are the same as in vertebrates with modifications. And so if we were to then compare the Hox genes in all of these different lineages, the question is, which ones were duplicated at which point, which ones formed the cluster, when were the entire clusters duplicated, because vertebrates have 4 Hox clusters when, say, fruit flies have 1. And so that's very important when we talk about genetics, but it comes up a lot when we start discussing the bilaterian higher groups. So I wanted to mention that, and I have some videos that go into it in greater detail, talking about the development of these Hox clusters. Okay, so last point to kind of bring all of this confusion to an even worse place is genetics can be a source of evidence to compare these lineages, but genetics isn't perfect, because your studies between long branch attraction, between mutations where genes can be present and then lost or not. So it sounds forward, and people looking at the evidence can still argue over, we need to do more studies because this isn't conclusive. All right. Well, how about then anatomical traits? Well, that is very confusing. Now, for example, you could say, oh, well, let's just use digestive traits, for example. You have complete digestive mouth and anus, or is there just one single opening? Is that single opening, did that develop from the embryonic blastopore, the first pore in the embryo, or no? Because then we could say, oh, you're a deuterostome because the first embryonic pore becomes the anus, mouth second, or a protostome where the blastopore becomes the mouth first. All right, so that's simple enough, right? No, because various lineages then have changed. Sometimes we think that the presence of one single opening of the digestive cavity is a primitive feature gotten from the ancestors, but in others, it seems that the ancestor of one smaller lineage had a complete digestive tract, but then lost it and now just has one single opening. What I said, the deuterostome development, where the anus develops from the blastopore. Well, that applies to some deuterostomes, but then in other deuterostomes, including the vertebrates, then the anus develops from another structure which forms. And here are some protostomes which developed as deuterostomes. And so then, once again, this is a good feature, which is useful, but not perfect. I know if we're trying to teach general biology, you'd like something in the textbook. Here is the one and only feature, to be considered here. But there are organisms which have evolved the absence of the digestive tract. There are other anal openings, not homologous to the anus, which developed in the first bilaterians. So digestive features are a problem. When did a brain develop? Well, we could argue maybe about, is every clump of nerve cells, is that a homologous structure? Like, is every cluster of nerve cells a brain evolved from the first brains? There are certainly features which are shared in the brains, whether it be the genes expressed, the midbrain, hindbrain boundary. So all of these things are shared between vertebrates, nematodes, arthropods, and annelids. More arthropods, I'm sorry, rather than nematodes. So these are things which are shared. So then that seems to imply that the last common ancestor of these lineages had these traits. That's why they're shared here. But then some of these other, groups might lack those brain features. So did that mean that their ancestors had them and lost them? All right, that doesn't make as much sense. Or did they evolve separately using the same genes? Also doesn't make as much sense. Where did hearts come from? Well, hearts can be different between, say, animals which have a closed circulatory system, where the fluids are always within blood vessels, or an open circulatory system like in a fruit fly, where the fluid can leave blood vessels and circulate in the coelom before returning back. But nevertheless, there are genes which are shared. Not only do human embryos, which are saying put a heart here, use certain genes, but fruit flies can use the same genes. So it is unlikely, not expected, that if hearts evolved more than once, that different organisms which separately evolved hearts would use the exact same genes to say, put a heart here. But that implies then that the heart evolved here. But that doesn't make as much sense because nematodes don't have a circulatory system, nor do flatworms. And so how is that explained? And the same thing then appears with these other traits. Arthropods have limbs, vertebrates have limbs. And they use some of the same genes, not to really make the limb per se, but at least to signal the body wall to say, put a limb here. Even some annelids have parapodia, which are using, so here's parapodia, not a true limb, but an outgrowth of the wall. So one could make the argument, if multiple lineages are using the same genes to signal put a limb or a parapodia here, then that must have existed in the common ancestor. But then again, some other members, like flatworms and nematodes, they don't have any trace of those limbs. Did they have them and lose them? While only the chordates have a notochord, some invertebrates have a structure which might be a precursor, where it has some of the genes expressed in the vertebrate notochord. There are examples of cartilage, which are known in arthropods, or cuttlefish, which are mollusks, or in a brachiopod. And so if the new organization of bilaterians is correct, then that's a problem. So then here's the issue. In general biology, we would like to just introduce these groups and say, here's how they're related. Here's the ones which are most closely related. Here's a biological group. It's hard though. All right. There's lots of evidence, but it doesn't always agree. And in science, that's okay. All right. Once again, science is a very gradual process, how we try to understand the natural world. And a lot of times it's difficult, and you have to revise models, etc. The original model held that flatworms were primitive, nematodes were primitive, and the animals which is coelom formed a group. And there's a lot of good anatomical traits which would support this. This makes sense when it comes time to considering the evidence of the heart and the segmentation and cartilage and those other structures, the brain, etc. So those anatomical features would tend to support this type of classification. But a lot of genetic sequences have been run which very strongly support this revised bilaterian classification scheme, where flatworms are relatives of mollusks and nematodes are relatives of arthropods. But now the anatomical evidence doesn't seem to match as well. So for a general biology student who was hoping for just tell me the answers that I can memorize, and then I'll know what every scientist agrees on, right? This has been a long and frustrating search for answers, and it's not over. The answers don't agree with each other. But once again, this is science first. And when you go into science, you need to understand this, and perhaps it would be you who would help clarify this with new studies, new hypotheses, and the like. And so this was meant to just offer some points on varying lines of evidence in the classification of the bilaterally symmetrical animals, the bilaterians.